Hey guys, how you going? Mason here. Most of you know me from Hot Copper, but if someone has been, falling down a rabbit hole in YouTube land and has fallen upon here, I'm here to talk about, RAC, Race Oncology, and look into FTO and Bisantrene. Uh, Apparently there was a lot of interest for this to happen, and I'd like to thank my very esteemed and very well credentialed manager Pex for throwing me in the deep end with this one. And while I can't say I'm 100% in my comfort zone here, just thought I'd give it a crack and sort of see how we go. So first thing is I want to just point out that I am 100% just your normal everyday guy, and with that I encourage everyone to do your own research and come up with your own conclusions. I am available. I've been documenting this stuff for a little while now, and I'm happy to provide some resources if that's what you're after, and we can get a bit of a chat going. It's cool. It's good science. Also, another point is we need to, when I'm referring to these processes or these mechanisms, I'm coming at it from an FTO overexpressing perspective, and that carries on through the presentation. So some of you may be like this, and that's okay. I've had my fair share of moments like that, and hopefully by the end of today we'll be somewhere like this. We've got this. Okay, so my learning objectives. So what I really wanted to focus on was the basics. What the hell does an FTO inhibitor? What is Bisantrene doing? Very, very simply. I then wanted to look at anti-PD1 therapy. And I wanted to try and explain its mechanism of action. Now, it might seem a bit weird doing a section of the presentation about this mechanism, but I think when I explain the mechanism and at the end there I compare it to FTO inhibitory therapy, it can help to understand and conceptualize, what an FTO inhibitor does compared to, another modality of anti-cancer therapy. So I think that's really important. We're going to explore Bisantrene's mechanisms of action. Now, they're not going to be all of them, but it will be some, and I will look at, comparing, anti-PD1 and FTO and seeing, how they synergize, how are they actually working together, and this comparison is one of the reasons why I'm so excited about the future of race oncology, and getting stuck into these trials. So very briefly, this is what an FTO inhibitor does. So this is the FTO protein. This was taken from the City of Hope, and more specifically, Bisantrene down here binds very tightly to the catalytic pocket. So it's a catalytic pocket on the FTO protein. So it just slips in there and it takes up that space. Okay. Now that might sound like gibberish. So if we imagine the FTO protein as a lock, and then what we're going to do is we're just going to this dark area here. So if this lock is an FTO protein, then this little pocket here is the catalytic pocket. And when Bisantrene comes in and it binds into that catalytic pocket. And that's all it does. And it prevents this M6A, and we'll go into a little bit more detail a little bit later, but it prevents this M6A molecule from entering into that catalytic pocket. And by preventing that from happening, there is a flow-on effect that has many different functions within the cancer cell, and actually other cells, but we'll find out more about that later. So that's pretty much it. So it comes in, binds into this catalytic pocket, and it prevents that FTO protein binding to that M6A. and then there's a downstream effect that that influences the expression or the production of genes, which influences proteins, which influences cell function. But I'll expand on that in a little bit. So I wanted to focus on anti-PD-1 therapies purely because they are an enormous market. So 20.1 billion in 2018, and they were projected to reach about 77 billion by 2026. And you know, they've had approvals in multiple different cancer types. I think there's 20 or 30 FDA approvals, and you know, it's the mechanism is found on many different cancer cells. So I thought that, you know, a anti-cancer therapy that is, you know, obviously worth a lot of money, I thought comparing to that would be good. So here we go. Now I'm going to try and keep this simple, MA 420, simple for you, MA 420. But there will be some concepts that are just a little bit complex, but I'll try and I don't have any Lego blocks, I don't think, so this will have to do. So this is I'll use my pointer. So this is an immune cell. So I'm going to discuss the anti-PD1, anti-PDL1 mechanism of action. But first I need to discuss what's actually happening with the interaction of the immune cell, the immune system, which is this CD8 plus T cell, and tumor cells. Okay, so first of all, what happens is this immune cell, this CD8 cell, binds to this tumor cell, okay, right here. And then what happens is, without going into too much detail, there is this cascade of events that come down here, okay? So that binding triggers the cascade of events. This cascade of events comes down into this section here, and what that does is that upregulates, or it increases the transcription of the PDL1 gene. Okay, now that gene is encodes for the protein PDL1. So basically what is happening is this binding is causing that cascade of events, which then leads to an increase in production of this PDL1. Okay, now it's expressed within the tumor cell and it moves from the cell up into the cell surface. Now this PDL1 is immune evasion. Pex wanted me to say Klingon. It's the Klingon cloaking device. So that's what it is. So obviously it upregulates this expression of PDL1 on the surface, and that makes it cloaked. And what happens is it under normal circumstances, without an anti-PD1 or PDL1 therapy, there is a linking of these two receptors. Okay, now when these two receptors link, the immune cell, this CD8 plus T cell, is being told, hey, look, we're one of you, we're one of the good guys, you know, we've got our cloak on us, we're not cancerous, don't come and get us. And so the immune system is essentially being switched off. What is happening when you use an anti-PD1 or PDL1 therapy is the molecule is coming in and it's binding to one of these receptors. Okay, and what happens when it binds to these receptors is it stops this interaction taking place. And in effect, what that's doing is it's revealing the cloak, and it's exposing that tumor cell, so that the immune system, this cell here, can recognize it as a cancer cell and go, hey, I know you, you're one of those Klingons, I'm going to come in and I'm going to destroy you. And that's pretty much it. So binding, cascade of events, PDL1 is increased, and that tells the tumor cell, the immune cells, we're one of you, and then the anti-PD1 kind of gets in the way of that and stops that from happening. So the immune cell can recognize that this is a tumor cell and go and get it. Okay, Bisantrene. So after doing quite a bit of research into this, I've the best way that I can describe bisentrene's mechanism of action is using a tree, and more specifically an inverse tree when you flip this upside down. And the key parts are the root, and then these different branches, okay, and then finally the leaves. And I'll explain this in a lot more detail later. Okay, so as I mentioned before, so we're keeping in mind that we're talking about cancers that overexpress FTO, that have a really high amount, and that high amount of FTO is leading to a poor prognosis. So what that means is the cancer is getting worse. You know, it's the cancer's progressing, it's getting worse, and it's because of the high amount of FTO, right? As I've said before, it binds to that catalytic pocket, and if we use our little lock example, that's in here, so it disrupts that binding between these two molecules. And what I didn't say is that it alters the expression of genes and proteins, okay? So by that not binding, it alters the expression of genes that encode for proteins, and then that influences cell cycle arrest, which I've called the brakes, apoptosis, which is deemed cellular suicide, immune evasion, which is that cloaking mechanism that I was talking about, and cellular metabolism, so energy. And for people that have been following me on Hot Copper, I've also been discussing autophagy, and there are other things that are influenced by the binding, by inhibiting FTO. Okay, so let's see how we go with this. This is a study done by knocking down FTO. So this CFTO is knockdown FTO. And this is really just to highlight the amount of genes that are influenced by this process. So genes downregulated by knockdown FTO equal 350. So what you have is by targeting a specific protein, and this we come back to this tree example. So by targeting one specific protein at the root of the tree, you can influence hundreds of different genes. Now, those genes encode for proteins, and those proteins have a function within the cell. So you're obviously going to be changing the way that the cell functions by targeting that one specific root, the protein. And so to put this into context here, so under normal cancerous circumstances where FTO is overexpressed, we have this binding, okay? And basically what the FTO is doing is removing this M6, okay? And it's producing a lot of this A, okay? So this interaction is happening a lot, and it's driving across and producing a lot of that A. Now, that has a flow-on effect to our genes, which then influences the proteins that are produced and the amount of proteins that are produced, and which influences cellular function, okay? And so what we have is bisantrene coming in here, disrupting the binding of these two molecules, which then leads to a reduction in this A and the accumulation of that A, which then influences the genes, which influences the proteins, and which changes that cellular function. And that is what we're going to discuss moving forward. All right, cell cycle arrest. So this is the brakes. So this is one of the mechanisms, excuse me. This is one of the mechanisms. It's the cell cycle. I'll explain the cell cycle first and what it all means. So we have this G0 phase, this G0, and this is our resting phase, okay? So the cell's kicking back, relaxing on a beach somewhere. It's probably got a stiff drink. It's not doing much at all. It's quite inactive. We have this G1, so this cell growth phase, and I'll just circle that real quick. We have this cellular growth phase. We have this DNA replication, preparation for mitosis, and this cell division, which I'll circle as well. And so what happens is when we have a cancer cell that is overexpressing FTO, leading to that poor prognosis, the cell cycle is sped up, it's increased, it's faster. For those that have been following me on Hot Copper, I've shared research that has shown that cancer cells that overexpress FTO have a really high amount of growth. They have this really high amount of proliferation, or what's also called cell division. And so what's happening is they're getting bigger and they're dividing faster. And that's where you get that increased growth, increased proliferation that is resulting from FTO, because it's increasing this cellular cycle and speeding it up. And it's doing this by modifying the expression of these genes down here, as we were talking about before. So FTO speeds up this process, right? And so now if we keep this model in our head, so bisantrene is blocking this and it's negating that, which is then influencing genes, proteins, and cellular function. So this is from the City of Hope. Now this DMSO, this is our control. So this is what we would consider as FTO overexpressing, or a normal cell. And so you can see that it does look a little bit confusing if you're looking at it for the first time. It kind of looks like I'm leading you down a garden path, really. So I'll explain it just quickly. So this red box here indicates G1, and we've got 3 G1 boxes here. So this is that cell growth phase. This blue box indicates these next 3 phases here, which is indicated here. And that G0 phase, which is our resting phase, is over here. And this is the gold box. And so what you'll see when we dose with CS1 bisantrene, compared to our control, you'll see that there's 3 times less cells that are in that G1, that red box phase. And what that means is that by inhibiting FTO in here, it has influenced the genes, proteins, and cell function by slowing down this phase of the cell cycle. Okay, so bisantrene has come in and it's reduced that cellular growth. So we see a reduction in growth. You'll also know that there's about a 12% reduction in this blue box here, which indicates this other section of the cell cycle, and one of those sections is that cell division. So we're seeing a reduction in DNA replication, preparation for mitosis, and this cellular division compared to when we're compared to our control, and this through this mechanism here. And lastly, you'll see that compared to the control, there's 56% cells, and that's roughly 40% increase in the amount of cells that are going into this G0 resting phase, kicking back, and they're basically inactive. And so what this is telling us is that when you dose bisantrene, it is arresting the cell cycle. It's putting the brakes on this process, and it's slowing down cellular growth, and it's slowing down cell division or proliferation. And it's doing it through this mechanism. One last point is when you start to compare bisantrene to Brequinar, you can actually start to see that, yeah, okay, there's more than half the cells in the G1 phase for bisantrene than there are for Brequinar. So that means less cells are going through this G1 growth phase. About the same are going through this phase, but there's about a 20% increase in the cells that are going into this resting inactive phase. And when you start to compare bisantrene to Brequinar in this way, you can start to see some of the ways in which bisantrene is superior. Okay, so apoptosis. So this is a kind of a funky word, and it basically means cell suicide. So if DNA is damaged within the cell, it will, in normal circumstances, it would mean that the cell undergoes this apoptosis or cellular suicide. I don't want to go through it in too much detail, but basically what happens is you've got a normal cell, and it realizes that something's wrong, and then it goes through a whole process of where it basically gets destroyed, right? And what's happening is, if we look down at this chart down here, and I'll circle these. Oh gosh. And so what you'll see is that we have, this is the apoptosis, so cell suicide, this process here, and we have 0 bisantrene. Okay, and you can see that there's not much apoptosis happening, and that's because the high amount of FTO is altering the genes to prevent the process of apoptosis, and it slows it right down. Okay, now what happens is, and you'll notice that as we start to increase the dose of bisantrene, you can start to see the rates of apoptosis, the percentage of apoptosis, this cellular suicide, start to increase. And I'll just draw that on the graph. Whoa. You can see that as the dose of bisantrene increases, the rates of cell suicide, so the cancer cells, bisantrene comes into the cancer cells, it's now being slowed down, it's got the brakes on for that cell cycle, and within the cancer cell, it's undergoing that cellular suicide, so it's going through this process here of breaking down and dying. And the way in which this is happening is this mechanism up here. So bisantrene is binding to that catalytic pocket. It's preventing M6A from turning into A. That doesn't happen, and that alters the genes, these genes down here, which alters the production of proteins, which alters cellular function. So instead of inhibiting cell suicide, it's now able to go through that process, which leads to a reduction of cancer cells. And that's pretty much it. So we have immune evasion. Now we have a lot of data from the City of Hope, and so I was going to focus on AML cancers, as well as the anti-PD1 cancers. So this is AML. So this is the expression level in AML, and these here are genes. So I'll keep this nice and simple. What you'll see is that there's a really high expression of this gene here, LILRB4, and this is a gene that encodes for a protein that modifies and regulates the immune system. Okay, so going over to this figure here, within a cancer cell that has a high expression, that has a high production of this LILRB4, it is going to bind, similar to the mechanism of anti-PD1, to a receptor on the T cell, so our immune cell. And it's going to be same thing. Hey, don't kill us. We're one of you guys. We've got our cloak on. And yeah, so don't kill us. And so it's preventing the immune system from activating and killing that cell. And so if we look over here, this is from the City of Hope, and I'll just draw here. And so if we look here, it's 100% expression, so normal expression, which is quite high. And then after you dose with bisantrene, you can see that expression level decreases quite significantly. And so what is happening within this cancer cell is you're having a reduction. You're having a reduction in this gene, which is limiting that gene's ability, which is limiting that cancer's ability to send out that signal to the immune system of, hey, we're one of you, don't kill us. And instead, that cloak is now being pulled off, and the immune system is able to identify that, yes, in fact, this is a cancer cell, and it can come in and, do its immune job, just come in and sort of kill some cancer. And that's pretty much it. And so this is PD1 or PDL1 immune evasion. So just give me 2 secs. I'll just highlight some things. So this up here is SHFTO is knockdown FTO. Okay, so this grey box indicates that the FTO has been knocked down, it's been reduced in concentration. And we want to focus on this gene, that gene right there. Okay, so what you'll see is this grey box indicates that FTO has been knocked down, it's been reduced within the cell. and that has had an influence on these genes down here. And you can see that FTO is at the end, and that relative to a normal cell, so normal cell NC, the amount of FTO has been reduced substantially. And this PDCD1 is important because this gene encodes for PD1. And you can see that after you have knocked down FTO, there is a reduction in the expression of that gene. Okay, so knock down FTO, similar to inhibition, you have a reduction in the gene that encodes for the protein PD1. Okay, the table below, this is for colon cancer, I believe. So you have PDL1, and you'll see that within the control cell there's 100% expression, and then they've used another knockdown FTO model. They've used 3 different ones, and you'll see that once you've knocked down FTO, the expression of PDL1 decreases quite significantly here on all 3 models. Now, this is great, but to put this into like wider context of anti-PD1 therapies, we will look back onto this schematic here of the interaction between an immune cell and a tumor cell. And so I'd just like to highlight what happens. So there's an interaction here, okay, and then there's this cascade of events that come down, okay. This cascade of events that come down, and under normal circumstances, if you remember, there's this upregulation of PDL1, okay, which makes its way out onto the surface of that tumor cell. Now, what we have is knockdown of FTO or an FTO inhibitor coming into this space. So it's coming into here, and it's blocking. You can see that it's blocking the production of this gene, and so what that means is it's blocking the transcription of the PDL1 protein, which limits the amount of protein that can be put onto the surface of the tumor cell and be a signal to the immune system of, hey, we're one of you, we've got our cloak on. So it limits this. But what's important is that, particularly in the context of synergy with an anti-PDL1 or PD1 therapy and an FTO inhibitor, is you can see that there is still some expression. Okay, so there are still some PDL1 being expressed up onto the surface of that tumor cell. But when you combine it with an anti-PD1 therapy, you then have those molecules coming in and binding to the top here and preventing. You can prevent that interaction, so then you have the immune system can then further identify tumor cells as cancerous, and then enable an immune response against that cancer. And this mechanism explains why, particularly me, I'm super excited to see an anti-PD1 therapy used in combination with an FTO inhibitor, because this is how they are going to interact together. Now, we are still in the infancy of understanding this science and the potential of this science. But up here we've got an anti-PD1 or anti-PDL1 therapy used in combination with DAC5-1. Now, this is another FTO inhibitor. And we can see that there is this massive reduction in tumor volume, so the volume of tumor. So you can see here it grows, and we use our DAC5-1, we use our anti-PDL1, and we can see this maroon line here comes right down, and to me it looks pretty much like 0. Now Bisantrene is 3 times more potent than DAC5-1 at inhibiting FTO, so it really starts to show the strength and the synergism between anti-PDL1 and DAC5 or an FTO inhibitory therapy. Unfortunately, my massive head is taking up the section of this graph to some extent, but this is in a melanoma cell, and you can see that the maroon line here is the combination therapy, and you can see that in comparison to, you know, just an FTO inhibitor or just an anti-PDL1, you can see that there's a reduction in that tumor volume when you combine these therapies together, and this mechanism is where RAC are heading right now, and it's quite exciting. Okay, so I didn't want to spend too much time on here. For those that are on Hot Copper, they've seen this before, but this by understanding what I've explained before, I imagine it makes it easier to understand how there could be more synergies with, you know, for example, tyrosine kinase inhibitors, alkylating agents, PARP inhibitors, because there's so many genes that are affected by the inhibition of FTO. Yeah. So the third mechanism that I really wanted to talk about was cellular metabolism. Now, this is our energy. This figure here, so these funky little lettered things are genes, and I'm going to enlist the help of Index Investor, and he's going to email out a quiz, and I'm going to get, you know, the RAC Hot Copper community to list all of the genes that I've mentioned here today in alphabetical order, if that's okay. But what back to what we're talking about. So this SH FTO is a knockdown FTO. There's two different types here, and basically what you're seeing is you have this normal expression up here, and that when you knock down FTO, you're reducing that FTO protein. Essentially what's happening here, you can see that it changes the expression of the genes, which would then influence the protein, which would then influence the cellular function. Now, that cellular function here is shown on this graph here. Now, this ECAR is a measure of energy. We'll call it energy just for simplicity. And you'll see that compared to the black line, which is the control, you'll see that after dosing with bisantrene, there's a massive, it's probably half, it's significant, but it's pretty much halved the potential of this cell to produce energy. So what's happening if we use this model, bisantrene is binding, preventing this interaction from taking place. An accumulation of this happens. It changes the genes, which are here, the expression of the genes, which influences the proteins, and it influences that function, that cellular function. Now, this is really important when we look back onto our synergy with anti-PD1 therapies. When we look at this figure here and then put that into context of this interaction here, so we can see that as tumor cell glycolysis, so cellular metabolism, energy production decreases, we see this upregulation of CD8T activation. Okay, so by inhibiting energy production here, glycolysis, you then activate the immune system, because both of those cells are looking for the same core component. And so when you knock out the cancer cell's ability to use that core component, more is left over for the immune cell. It's pretty much like you've got two groups and they're at the same bar and they're trying to eat all the chips. And so FTO is stopping the cancer cells from getting the food, and then the CD8 T cells can pick up the food and have the energy to activate and you know, exert their effect. And so if we put that down to this graph here, and I'll just run through it all again. I feel like repetition is the best. So there's this interaction here. We then have this cascade of events. Okay, our FTO inhibitor is coming in here, and our anti-PD1. So we have this cascade of events. FTO inhibitor slows this transcription down, reduces our PDL1. The anti-PD1 or PDL1 therapy blocks this interaction, so the tumor can recognize this, the immune cell can recognize this tumor cell for what it is, Klingon, I think. But then you also, when you consider cellular metabolism, is you can actually appreciate that there's also, it's not exactly like this, but there's also this transfer of energy where the tumor cell is losing energy, the ability to produce energy, and the immune cell is becoming more active. And understanding this concept is really important when you bring an FTO inhibitor and an anti-PD1 or PDL1 therapy together, because there are these synergisms and these interactions. Okay, so I have to remember what sequence I've put all these little figures. So this is our inverse tree, and I think index investors going to really like this slide for some reason. Hopefully I get 2 thumbs up from him. So at the top here we have, this is our inverse tree, and at the top here we have this interaction. So there's no bisantrene, so FTO is overexpressed. These cells interact, these molecules interact, and it produces a lot of that. So it comes down the tree and it influences the cell cycle. It increases the cell cycle. It decreases apoptosis. It essentially puts the accelerator on. You can see that I've explained that it puts its little cloak on, so it can't be seen by the immune system, and it has this massive increase in energy production. And so what we have is when bisantrene comes into the mix and it stops this process from taking place, we then see apoptosis is left out. We see the brakes put on this cell cycle. We see cellular metabolism decrease, so the production of energy decreases. We see this reduction in immune evasion. We see cancer cells becoming more sensitive to anti-cancer treatments, and we see apoptosis, or this cellular suicide, increasing, this death of the cells. And this figure illustrates it the best. So it comes down into each of these branches, and each of these little leaves is thinking about it like a protein. So changes up here, comes down, and each of these sections relates to a different function within the cell. So you have this change up here, which influences the genes, which influences the proteins, which then influences the function of the cell. And I'd just like to point out this set of leaves down here. So if we think about these leaves as PD1 or PDL1, then we know that those leaves are worth about 77 billion US, or they expected to be worth US 77 billion by 2026. And I think when you start to look at the model in this way, in this inverse tree way, and you start to appreciate, what kind of effect you can have by targeting the core or the root of these mechanisms, you can start to appreciate maybe why I am very excited. I personally am very excited about bisantrene in general and Race Oncology. Because I think when you start to actually understand that these 2 leaves down here are worth 77 billion by 2026, then you can start to appreciate the value of targeting this mechanism. And I think it also makes sense why so many different researchers and research groups are investigating this science, because of it's central to not only cancer but so many other diseases out there. And as I said, by targeting this mechanism, you can influence 350, just hundreds of genes that has an effect on the cell. And that's pretty much it. So I hope that was useful. This is my first one. I don't know if you guys really want to have another one. But yeah, that's pretty much it from me. On a more personal level, I'm very excited to see where this science goes. It's very, very compelling for me, because I feel like every new piece of research that comes out is another puzzle piece. And I'm very excited to see where this, where the science leads, and really starting to see some potent therapies for people suffering with cancer. And yeah, just shout out to all the hot copper guys. You know who you are, and it's a pleasure to go through this with you. And yeah, if you guys have any comments or queries, let me know. And yeah, I'll see you on hot copper sometime. Sweet as. Cheers, guys.